Welcome back to G Fun Facts Online. Today, we are not just talking about a scientific breakthrough, we are talking about a fundamental rewriting of the rules of biology. You have heard of CRISPR. You know it as the genetic scissors that changed the world in the 2010s. It earned a Nobel Prize and ushered in the second era of genetic engineering. But today, we are discussing what is being called the third genetic revolution. It is a discovery so elegant and so powerful that it makes our current tools look blunt by comparison. We are talking about Bridge RNA. For billions of years, a secret war has been waged inside the genomes of bacteria. It is a war of information, fought with code. On one side, you have the hosts. On the other, the invaders, viruses, plasmids, and transposable elements. It is in the debris of this microscopic battlefield that humanity has often found its most powerful tools. This is where we found restriction enzymes, the scissors that launched the first era of genetic engineering back in the 70s. It is where we found CRISPR-Cas9, the guided missile that allowed us to edit the book of life. And now, from those same murky depths of microbial evolution, a new tool has emerged. Discovered by a team at the Arc Institute led by Dr. Patrick Farshu, Bridge RNA represents a shift from a clumsy cut-and-paste operation to a sophisticated, programmable word processor for the genome. To really understand why this is such a big deal, we have to look at the history of jumping genes. Back in the 1940s, Barbara McClintock discovered transposable elements in maize. These are segments of DNA that spontaneously move from one location in a genome to another. For a long time, scientists dismissed these as junk DNA or selfish parasites that just cluttered up the genome. But nature rarely keeps junk. These mobile genetic elements are actually engines of evolution. They shuffle genetic decks, creating diversity. Among the most fascinating of these is the IS-110 family of insertion sequences. Most jumping genes are destructive. They rip themselves out of the DNA, leaving jagged scars, or they copy-paste themselves in a way that relies on the host cell to fix the mess. But the IS-110 element is different. It is clean. It is precise. Dr. Su and his team were scanning bacterial databases looking for nature's hidden editors. They weren't looking for another pair of scissors like Cas9. They were looking for an architect. They focused on IS-110 because it had an enzyme called a recombinase, which can rearrange DNA strands seamlessly, but it seemed to lack the complex proteins usually needed to guide it. The question was, how did IS-110 know where to go? How did it navigate the complex library of the genome? The answer, published in Nature in mid-2024, is Bridge RNA. Here is the mechanism, and this is the part that is truly revolutionary. Unlike CRISPR, which uses a single guide RNA to find a target, Bridge RNA is bispecific. It has two distinct hands, or binding loops. Imagine the Bridge RNA molecule as a specialized robotic arm. One hand, called the target loop, grabs onto the specific location in the host genome where you want to make an edit. The other hand, the donor loop, grabs onto the DNA sequence you want to insert. This is the molecular handshake. CRISPR is like a blowtorch. It cuts a hole in the hood of your car, that is, the DNA brake, and hopes the mechanic, which is the cell's repair machinery, can weld a new engine in before the car falls apart. Bridge RNA is totally different. It holds the chassis with one hand and the new engine with the other. Then, catalyzed by the IS-110 recombinase, it performs a synchronized swap. It unbolts the chassis and bolts in the engine in a single, fluid motion. There are no double-strand brakes. There is no reliance on the cell's shaky emergency repair crews. It is a self-contained, all-in-one editing system. This brings us to why Bridge RNA is a paradigm shift compared to CRISPR. CRISPR works by violence. It slashes through the DNA double helix. The cell perceives this as catastrophic damage and rushes to fix it. While scientists can provide a template for the cell to copy during this repair, the process is error-prone. It can lead to indels, which are random insertions or deletions at the cut site. It can cause translocations, where large chunks of chromosomes break off and attach to the wrong place. It can even trigger p53, the cell's guardian protein, causing the cell to commit suicide because it thinks it is dying. Bridge RNA avoids all of this. The recombination reaction is concerted. The DNA is held together by the enzyme throughout the entire process. It is scarless editing. Then there's the payload limit. CRISPR is great at breaking genes or changing a single letter, but it struggles to insert large sequences. If you want to insert a whole gene, say a healthy copy of the CFTR gene for cystic fibrosis, CRISPR is incredibly inefficient. Bridge RNA, however, evolved to move entire transposons, which can be thousands of bases long. It is naturally adept at handling massive payloads. In their initial experiments, the team successfully inserted payloads of over 4,000 base pairs with high efficiency. Theoretically, it could insert sequences vastly larger, essentially allowing us to upload entire gene clusters or synthetic circuits into a genome. Another massive advantage is the programmability of the donor. In CRISPR, the guide RNA only tells the enzyme where to cut. The donor DNA is just floating nearby, hoping to be picked up. Bridge RNA is the first system where the donor is also programmable. The donor loop on the RNA molecule specifically recruits the DNA you want to insert. This gives scientists control over both sides of the equation, the destination and the cargo. This modularity essentially turns Bridge RNA into a word processor. By simply changing the sequences in the two loops, scientists can program the system to perform three distinct operations. First is insertion, or paste. This takes a foreign piece of DNA and slots it seamlessly into a specific location. This is what you would use to replace a broken gene with a functional one. Second is excision, or cut and delete. By programming the loops to recognize the two ends of a sequence already in the genome, the recombinase can bridge them together and snip the middle section out. And third is inversion, or flip. If the target sites are oriented in opposite directions, the recombinase will flip the DNA sequence between them upside down. While that sounds abstract, it is a powerful tool for gene regulation, potentially creating biological switches where a gene is turned on or off by physically flipping its promoter sequence. So what does this mean for the future of medicine? We are potentially moving from treating genetic diseases to curing them at the source. One of the most promising areas is repeat expansion disorders. Diseases like Huntington's disease and ALS are often caused by stuttering DNA short sequences like CAG that repeat dozens or hundreds of times. These repetitive regions are toxic and incredibly difficult for CRISPR to handle, because the scissors often get confused and cut in the wrong place among the repeats, causing genomic chaos. Bridge RNA offers a precise solution. It can be programmed to bind the unique sequences flanking the repeats, cleanly excising the toxic stutter and stitching the healthy ends back together, effectively restoring the gene to its normal length. Beyond fixing broken genes, we are looking at next-generation cell therapies. Right now, CAR-T therapy, where immune cells are engineered to fight cancer, relies on viral vectors that insert DNA randomly, or CRISPR, which is inefficient for large insertions. Bridge RNA could allow for the safe, specific insertion of massive, complex synthetic gene circuits into immune cells. We could engineer cells that not only kill cancer but have safety switches, logic gates that only attack if multiple conditions are met, and cytokine factories to boost the immune response, all programmed into a single DNA cassette and inserted with Bridge RNA. And it is not just medicine. This is a holy grail for synthetic biology. In agriculture, we might want to engineer crops that are drought-resistant or nitrogen-fixing. These traits often require not just one gene, but whole pathways involving dozens of enzymes. Bridge RNA's ability to move large chunks of DNA makes it possible to stack these traits efficiently. It opens the door to whole genome restructuring, where we could streamline bacterial genomes to create cell factories that produce biofuels or bioplastics with maximum efficiency, removing millions of bases of unnecessary junk DNA and reorganizing the rest. However, we have to be realistic. This technology is in its infancy. The jump from a test tube or an E. coli bacterium to a human cell is significant. One major challenge is delivery. Getting the large recombinase protein and the complex bridge RNA structure into the nucleus of a human cell is a hurdle. While lipid nanoparticles have worked for mRNA vaccines, the specific stability requirements for bridge RNA in human cells need optimization. Then there's the issue of chromatin. Bacterial DNA is naked and accessible. Human DNA is wrapped tightly around histone proteins. We do not yet know how easily the IS-110 recombinase can access these closed, tightly packed regions of the human genome. And of course, there is safety. While theoretically safer than CRISPR, deep sequencing studies will be required to ensure the target loop doesn't accidentally bind to a similar-looking sequence elsewhere in the 3 billion letters of the human genome. To wrap this up, think about the trajectory we are on. Generation 1 gave us zinc finger nucleases and talons, tools that allowed us to edit genes but were difficult, expensive, and slow. Generation 2 was CRISPR, which democratized gene editing, making it cheap and easy, but lacked finesse for large-scale architecture. Now we are looking at Generation 3, Bridge RNA. It promises to unite ease of use with absolute architectural control. The discovery of Bridge RNA reminds us that the most advanced technologies on Earth were not invented in Silicon Valley, but in the primordial soup of the microbial world. By harnessing the mechanisms of jumping genes, we are finally learning to speak the language of DNA fluently. We are no longer just cutting words out of the book of life. We are learning to write new chapters. As research accelerates, the next decade will likely see Bridge RNA moving from the lab bench to the clinic, offering hope for genetic conditions that were once deemed untouchable. Thanks for listening to Fun Facts Online, and we will see you in the next deep dive.